Hello again and welcome back to the last part of our lecture today on the SEM. And this part is rather short. It's on the extensions of structural equation models to generalized linear models, linear mixed effect models, and generalized linear mixed effect models. As an example, we will use this study published in Ecology, which was done in the beautiful place in the world called Serengeti. So the study aimed to understand where are the hotspots of animals in Serengeti, of animal biodiversity, and what are the factors affecting these hotspots. And on the first picture, you see the Serengeti that you may think of if you watch a video about this beautiful place. So we think about Serengeti as a place full of animals. And in the second picture, you see the place without any animals, so empty place. And the idea is that it's true, Serengeti is look like on the second picture, but there are hot spots of biodiversity of herbivores. And the idea of the study was to understand why these hot spots appear and what controls them. And here is a hypothetical model of the study. And hotspot is a main response variable here, which shows us whether these study plots are hotspots of biodiversity or are not hotspots of biodiversity. And authors presume that position in landscape affects whether this place is hotspot or not, because if position is further away from river and it's flat, it lowers the predation risk, and then it affects these places to be hotspots. Or also simultaneously other factors affect that, such as biomass of plants. Also, if we have low biomass, this helps predator avoidance. And if we have low biomass, it also improves the forage quality of plants. And if they have higher quality, it means that lower CN ratio in the biomass, then animals benefit from this. Now let's look onto the hotspot variable. So we can see here that this is binary variable, and I just plotted the effect of biomass on hotspot here, predicted effect, and we see that 0, it means here that no, this is not a hotspot, and 1 means yes, this is a hotspot of biodiversity. And what we see here that biomass, increasing biomass actually decreases the probability of the place being hotspot, because as we remember, the authors assumed that high biomass would prevent animals to see the predators there. So let's take this example in the piecewise CM in R. For this, you have a data on Blackboard called Anderson CSV file. And if you check the structure of data, you will see which them are numerical, and that hotspot is simply the level 0 and 1. And then, of course, we have here also the effect of biomass on leaf nitrogen. So we want also to specify this submodel here. But of course, we have to plot the QQ plots in order to see whether we can assume the normal distribution here of residuals. I would say that these are okay. We have a little bit deviation from constant residuals here, but this is not so bad because the data set is not so large here. So we presume that we can use the linear regression here. And because our hotspot, yes or no, let's say here, is binary, we use generalized linear model here with a family binomial, so as you remember. We will not go through details about the generalized linear models and the generalized linear mixed effect models and mixed effect models, as you have learned all of this in the previous 3 days with Felix. So We can check the summary, of course, if you want. If you would use some other family here, not binomial, but let's say Poisson or others, so if this would be rather not binary data, then we would check also for overdispersion, of course. But as this binary data, we cannot have the overdispersion here. So, and then if you are okay to go, we can use piecewise CM. And for this, we use our function PSEM and then call the summary. And what summary gives us? It gives us submodels, as you remember, AIC. Also, it gives us the test of directed separation. So this test suggested the missing link from landscape to leaf nitrogen, but this effect is not significant, so we will not consider this in our model. And we got our chi-square and Fisher-C statistics, and we see that the p-values are very high, so we can say that our model fit data well. And here you get the coefficients for each effect that we tested. You can see that all our effects are strongly significant. So in addition to raw coefficients, we also have standardized coefficients, and it's very new in piecewise CM, because of course when we have generalized linear models and we also compare them with linear regression models, then standardization is not so straightforward. So John Levchik did a huge deal of work in order to implement the standardization for the generalized linear models. And also we have the R-squares, which show how much variance is explained for each response variable here. And here on this example, you can see the final figure and the results. So I filled in the coefficients, and we can see that our model fits data well. And also we see that all effects are significant, and all hypotheses behind the assumption of the author are confirmed by this model. By this example, I wanted to show you that the implementation of generalized linear models, as well as mixed effect models and generalized linear mixed effect models, is very straightforward in piecewise CM. As far as you know how to deal with generalized linear mixed effect models and generalized linear models, then you know how to deal with them using CM. But for this, of course, you have to follow the procedures that you have learned in the previous lectures for each separate submodel. So you first have to follow the protocols which Felix gave you to test each submodel separately. And only after that, when you are sure about your submodels, you can bind them together in the piecewise CM. Given that last 3 days you had a great overview about the mixed effect models and the generalized linear mixed effect models, I will not show you in this part of the lecture how to use them in the CM, because as you understand, you simply first have to feed them separately in each submodel and then bind them in the piecewise CM, as it's very straightforward procedure. But I would ask you to do this task 3, which implements the mixed effect and generalized linear mixed effect models. So for this, we use data of Bill Shipley. And Shipley, as you remember, the scientist who created this local estimation procedure in the CM. So this data, Shipley, they are part of the piecewise CM package, and you can simply load it by loading a library of piecewise CM. And data are based on the predicting latitude effects on survival of a tree species. So here you can see the latitude, and here you can see live variable, which is a survival of tree species. And these are repeated measurements on 5 trees at 20 sites. And these are done through time. So you have date here from 1970 to 2006. And variable live It's binary, which is 0 or 1, alive or dead. And it's influenced by phenology, which is the degree days until bath break. Um, and degree days are this variable dd here. And here again, you see all these variables, and take care because you have sites here, trees here, and you will notice if you think about data, and when you see the next slide, you will understand that we have different hierarchy here. We have different mixed models. So some variables are nested within other variables, and it's differ across our submodels. Here on this slide, I show you the separate submodels which you have to string together in the piecewise SEM package. So here is the first submodel where your DD variable affected by latitude, you have this piece, but note, think about the mixed effect here in each submodel. And the next one, date, is affected by DD variable, growth is affected by date, and variable live is affected by growth. And your task is to use these submodels in piecewise CM as a part of the CM model shown here above. And you have to think about two things here. So first, think about study design, why these mixed effects here are shown like as they shown here. And then do all the procedure in the piecewise CM, look through the data, through the results, and try to explain the results. Thank you again for your attention and looking forward to see you on the seminars.